Let us understand bioinformatics in little more detail. As I've already told, bioinformatics is collection, databasing, understanding, analysis, interpretation, and prediction of biological information for the welfare of mankind. Actually, it is a combination of two different branches, that is computer science and biology. In fact, it is application of techniques from computer science to solve the problems of biology. The problems that computer science generally solves are massive quantitative data related problems, desperate need for efficient solutions, and if you do so, you get success without any issue, without any problem, and with much ease. And the cell in turn is made up of chromosomes, and the chromosomes are made up of genes, and the genes are made up of DNA, and the DNA is made up of nucleotides, which are adenine, thymine, cytosine, guanine, and uracil, ATGC or UCAG. And the fundamental principle of bioinformatics is understanding the details of these sequences which are hidden in the DNA, which are hidden in the chromosome, which are hidden in the cell, and understanding it in deep manner, understanding it deeply, and getting the proper or correct information out of it. For example, what you're seeing on the screen is the chromosomes, and a deep analysis of this genes gives us huge amount of sequence data, and this huge amount of sequence data is enormous. For example, a single human cell stores 3.2 billion sequences, and this amount of sequences is humongous. For example, if you want to write all the sequences of a human genome on paper, it runs to about 175 books, and it runs to about 2,62,000 pages. That is one cell and genome present in one cell, that is about 3.2 billion sequences. So to understand this mind-boggling information is very difficult. To comprehend this information, comprehend the humongous amount of sequence, you require the tools that are there in information technology. Not only to understand this, to get some meaningful information out of it, you read bioinformatics tools, and that is what is the role of bioinformatics. It's humanly impossible to locate the genes in this long sequences of DNA, and bioinformatics tools will help us locate the genes. It's a very simple. The basic principle behind understanding this is knowing the unknown from the known. So our mind intrinsically, whether we like it or not, has a practice of knowing the unknown things by comparison. If you look at a long rope, a long structure which is wired or which is wavy, your mind tries to compare this with a rope or a snake. So finally, by comparing it, comparing it with the similar objects, the mind will finally come to a conclusion and decides what it is. So for doing a comparison, complete data is required, and hence all the data that is generated in bioinformatics is stored in databases. From this data, meaningful information is obtained. So you conclude finally whether it is a rope or a snake by comparison only with the help of complete knowledge. That is what your mind does. Similarly, even in bioinformatics also, if you have an unknown sequence, the unknown sequence is compared with the known sequence which is there in the database. You are seeing a tool which is a sequence similarity search tool. We have sequences in the database, and these sequences are compared. The unknown sequences compared with the known sequences, and thus you conclude what is the correct nature, correct meaning of the sequence that you have submitted. So this is one example of bioinformatics, and this knowledge is used in the understanding of a number of enigmatic diseases, like for example sickle cell anemia. About 3 lakh babies in the world are born every year with sickle cell anemia, and the difference between normal cell and sickle cell is very little. There is only one change in the amino acid, only one amino acid change, that is valine, okay, is present instead of glutamine, and that is also because of one change in the sequence, that is adenine instead of T. So adenine thymine replacement is causing a disease called sickle cell anemia, and this can be easily understood by bioinformatics, and the data, the information that is obtained by the bioinformatics can be used in order to understand, in order to solve the problem. So this is done basically by DNA sequencing. DNA sequence is knowing each and every letter of the genome or gene in detail, and it's an elaborate technique, and one of the important tasks of bioinformatics is sequencing. There are different methods of sequencing. As of now, 31,691 genome projects have been completed, and there are many more that are in the pipeline, which are being sequenced. Similarly, we are also able to understand the complete structure of a protein in order to understand its function, in order to modify these functions. So bioinformatics tools will help us understand complex genome, also complex proteins, in order to improve the life of a human being. So there are a number of popular tools that are available in bioinformatics, and some of the tools I'm showing it on the screen. And another important task that is there in bioinformatics is drug designing. You can design a drug, that is required for a particular disease. And two important achievements, two important breakthroughs of bioinformatics. One is the Human Genome Project, completion of Human Genome Project, which took about 13 years. It started in 1990 and completed in 2003, and this started a revolution. Now, as I mentioned earlier, many genomes have been studied. Similarly, one more technique which is considered to be one of the top 10 greatest inventions of the century, that is CRISPR, which is gene editing tool. Like I mentioned, in case of sickle cell anemia, if there is a defect in the gene, and the gene can be corrected in vivo. But this is one of the biggest achievements of bioinformatics. So apart from this, there are many other branches, many other applications of bioinformatics. We will study all these things in this course.